The process of urine formation requires filtration, tubular reabsorption, and tubular secretion. So why do we need reabsorption? Why is that a necessary mechanism for urine formation? Well, we're going to use reabsorption for a couple different things. Primarily, we use it to save any of the good stuff that may have made its way into the tubular fluid. When the process of filtration occurs, it's only size selective for the most part, and it also selects things based on their water solubility. So as long as you're small and water soluble, you'll make your way into the filtrate, even good stuff like glucose and amino acids. So we use the process of reabsorption to absorb back these nutrients and other substances. One example is glucose. We use active transport mechanisms to bring glucose out of the filtrate back into the peritubular blood. Technically, this is a form of active transport called secondary active transport, which requires co-transport with sodium. We'll talk more about this later. Another reason we perform tubular reabsorption is because sometimes we will reabsorb a substance in order to exchange it for something that we want secreted. So we link the process of reabsorption and secretion. An example of this is sometimes we will save sodium in exchange for secreting hydrogen ions, which are acidic. In other words, we want to secrete the acid, the hydrogen ions. And one of the ways we can do this is by exchanging it for the sodium ion. So we reabsorb sodium while secreting hydrogen ions. We also reabsorb some substances because it will actually help us to reabsorb another substance. An example of this is that we will reabsorb some urea. A lot of urea will make its way into the filtrate. And that's good because we want to get rid of urea. It's toxic. It's not good to have in our blood, which is kind of weird when we think about the fact that we do actually reabsorb some of the urea. But the reabsorption of urea is necessary because it will act as an osmotic agent that will help us to reabsorb water. Which parts of the renal tubule are involved with reabsorption? Basically, all of the renal tubule is. If you look at this picture here, the boxes that are sort of the pale purple, those are showing you substances that are being reabsorbed. And if you look, you can see that all of the renal tubule is participating. However, the proximal convoluted tubule is by far the best at reabsorption. It will reabsorb the most diverse amount of substances as compared to any other part of the renal tubule. What sorts of substances are reabsorbed the most? Nutrients such as water, glucose, amino acids, proteins if they happen to make their way in there, water-soluble vitamins. Other things are reabsorbed too, but maybe not at the same rate as like glucose. Examples include minerals and ions, especially sodium. We do a lot of reabsorption of sodium. We also reabsorb quite a bit of bicarbonate and quite a bit of urea, even though we don't want the urea, but we do need to reabsorb it because it helps with the reabsorption of water. Why do we need tubular secretion? Why is that important? Tubular secretion helps us to get rid of any bad stuff that's still in the blood that was not filtered. The filtration process is not 100% effective, and that means there are still waste and toxins that remain in the blood that we want to get rid of, and we can use secretion for that process. One of the things that we commonly secrete are drugs. I find the process of drug secretion really fascinating, to think that we have evolved mechanisms in our body to secrete these drugs. We have to have transporter proteins in the cell membrane of our renal tubule cells in order to move these drugs into the tubular fluid. So in this image here, we have something called OAT4 and OAT1. And these stand for organic anion transporters. And what this means is that these are proteins that are capable of transporting negatively charged anions that are also organic. And it turns out there's a lot of stuff that gets into our body that's maybe toxic or unwanted that we need to get rid of, and we can use these OAT transporters to do that. It also just so happens that a lot of drugs fall into this category too. Drugs like penicillin, they are anionic and they are organic, so they can be removed using these OAT transporters. These are active transporters, and it actually takes energy to do this secretion process. So the drugs like penicillin will leave the peritubular capillary through bulk flow, but then they'll bind to an OAT transporter. In order to secrete drugs, the drugs have to move out of the peritubular capillary through bulk flow, and then the drug has to bind to a transporter on the basolateral side of the renal tubule cell, where it will be transported into the tubule cell. And then it has to be transported out of the tubule cell into the renal tubule lumen by using another transporter protein, in this case OAT4. At this point now, the drug will be in the lumen of our renal tubule, and then it will remain in there and then end up being excreted with our urine. When individuals have highly impaired renal function, sometimes drug dosing has to be adjusted. Because of the impairment, these individuals might not be secreting drugs like they should. In other words, they're retaining more drugs in their bloodstream than normal. Because of this, their dosing might need to be adjusted downward. Now, we also secrete substances in order to exchange them for something we want to reabsorb. And we kind of talked about the same thing with tubular reabsorption. So this is like a two-way door, if you will. For example, sometimes we exchange bicarbonate for chloride ions. And what we're trying to do here is we're trying to save the bicarbonate for purposes of buffering, but in the process, the chloride ions are being sacrificed. So this is, again, another example of that linked reabsorption and secretion. Which parts of the renal tubule are involved with secretion? Just like with reabsorption, all parts of the renal tubule are involved. In this image here, the pale yellow boxes are representing substances that are being secreted. As you can see, the proximal convoluted tubule not only is our most diverse reabsorber, it's also the most diverse secretor. What substances are secreted the most? Metabolic waste like urea, uric acid, urobilinogen, and ammonia are very highly secreted. But we also secrete a lot of hydrogen ions, in other words, acids, and we also secrete some minerals, especially potassium and chloride ions. And again, we're secreting those because we're trying to exchange them for something we want reabsorbed. And of course, like we talked about, we also secrete a lot of drugs. I want to emphasize the importance of exchange in reabsorption and secretion. So we talked about that linked reabsorption and secretion process, where we're reabsorbing something while secreting another thing. These require membrane proteins like exchange pumps. In the picture here, we can see that embedded in both the basolateral and luminal membranes are sodium-hydrogen exchange pumps. These are active transporters. They require ATP. On the basolateral side, we are going to be pumping the sodium into the interstitial fluid in exchange for bringing the acid into the renal tubule cell. Then on the luminal side, we are going to be pumping the hydrogen ions into the tubular fluid in exchange for bringing the sodium ions into the renal tubule cell. And this process is really important for getting rid of acids. Besides the sodium and hydrogen ion exchange, there are some other important examples of linked reabsorption and secretion. Bicarbonate and chloride ions are another example. Like sodium and hydrogen ion exchange, we will use the bicarbonate and chloride ion exchangers during acidosis. During acidosis, the pH of the blood is too low, so we need to get rid of acids, as in the case of the sodium hydrogen ion transporters, but it also helps to reabsorb bicarbonate because bicarbonate can buffer the acid in the bloodstream. So another process that occurs during acidosis is we will secrete chloride ions into the tubular fluid, and in exchange for the chloride ions, we are bringing bicarbonate ions back into the renal tubule cell and then eventually back into the blood. Another important example of linked reabsorption and secretion is with sodium and potassium. This linked reabsorption and secretion of sodium and potassium occurs at high levels under the direction of the hormone aldosterone. When aldosterone is released, it will cause us to reabsorb sodium by sacrificing potassium. And this process is important because when the sodium is reabsorbed, water is also reabsorbed by osmosis. So whenever we save sodium, we are also saving water. But as you can imagine, this could become problematic because if it continues to happen and we continue to secrete this potassium, it can possibly lead to hypokalemia, which could cause problems with any cells in the body that depend on transmembrane potentials, like our neurons or our cardiac muscle cells. I also want to emphasize the importance of glucose reabsorption. We reabsorb 100% of the glucose that has made its way into that filtrate and tubular fluid. And we are able to reabsorb glucose by using a type of active transport called secondary active transport. Secondary active transport is actually pretty interesting. What we're going to do with secondary active transport is we are going to establish a diffusion gradient for one molecule, and then we are going to let that molecule diffuse, and as that molecule diffuses, it will bring along with it glucose. In other words, the glucose is going to kind of ride the wave of diffusion of this other molecule. And we usually do this with sodium. So here is going to be a cell membrane, and this is going to be my sodium pump. And I'm going to use the sodium pump to move the sodium out of the renotubule cells and put it into the renotubular fluid. So I'm going to build up this really high concentration of sodium ions in that renotubular fluid. Now that I have that high concentration of sodium in that renotubular fluid, I can use that sodium diffusion gradient to help bring glucose back in. I need a second transporter protein to perform this. And this is going to be a transporter that's going to allow for the diffusion of sodium. So this will be facilitated diffusion of sodium back into our renotubule cell. And as that sodium comes in, the glucose is just going to be real sneaky. The glucose just kind of like grabs a hold of the sodium, and then the glucose comes in too. So there might not be a diffusion gradient for glucose to come in, but it's just being really sneaky and using that wave of diffusion of sodium to make its way in also. This is considered active transport because this beginning process here, this requires ATP, and we are moving the sodium against its concentration gradient. So that's how we define active transport, right? Moving a substance against its concentration gradient and using ATP to do that. So this beginning part requires ATP, but the second part here doesn't. So that's why it's kind of a little bit tricky in terms of identifying it as active transport. I also want to emphasize the importance of water reabsorption. We reabsorb about 99.5% of all of the water that enters the nephron. We approximate that 180 liters of water, so that's 92 liter bottles worth, right? 180 liters of water make its way into the filtrate in one day. And yet we only have urine output levels of about 1 liter, which means we are reabsorbing 179 liters of water, and that is just a gigantic amount. So the nephron is an excellent water reabsorber. One of the important places this is occurring is in the nephron loop. The nephron loop has a lot of specializations that allow it to reabsorb a lot of water. We're not going to go into the details of how the nephron does this, but I just want to mention that the nephron actually intentionally creates an osmotic gradient around the nephron loop, and that osmotic gradient is used to reabsorb water. And this osmotic gradient involves urea, which is why we have to reabsorb some urea. The collecting duct also plays a major role in water reabsorption. And the collecting duct is really important because this is towards the end of the nephron, and this is sort of like our last chance to do any adjustments that we want to with our tubular fluid. So the collecting duct plays a major role with adjusting the final volume of water and hence the osmolarity of urine. Antidiuretic hormone and aldosterone are two hormones that can affect our water reabsorption process. And they can affect this collecting duct and the distal convoluted tubule. So they're there at the end to kind of do some final readjustments. Both antidiuretic hormone and aldosterone will cause us to reabsorb water, which will reduce the urine volume.